There we go. It's recording. People are going to start entering. Great. Well, hello, everyone. Thank you so much for joining us this afternoon for this very important webinar on teaching students with visual impairments about COVID-19, which is all on our minds. And it's extremely important for us to be able to provide this life-saving, in many ways, information. So that our kids have the same access as other children to learn about the coronavirus and how it affects our lives. My name is Olaya Landa-Villard. I'm the director of the APH Connect Center, which is made up of 3 different websites, Family Connect, Vision Aware, and Career Connect. And we also have the information and referral line as well. So I'm going to be here moderating and kind of hosting the webinar, but I'm definitely not the star of the show. The stars of the show will come up in just a little bit, but I would really like for the project leader for the development of this, of the health education guide to go ahead and introduce herself.
Hello, everyone. Good afternoon. My name is Roseanne Hoffman, and as Olaya said, I'm the STEM project leader, and I led the development of the health education for students with visual impairments guidebook for teachers. And I want to point out that one of the authors and one of our presenters today, Dr. Tiffany Wild, won a very important award, the C. Warren Bledsoe Award for 2020, which recognizes noteworthy pieces of literature in the VI field. And it's bestowed by the AER Awards Committee. And it's, it's important to recognize that it's one of the highest awards in the VI field. So we just want to make sure that Tiffany gets that recognition. And I think I will hand it over to Tiffany and Sonia, who will give the presentation. Thank you.
Well, thank you. And thank you for having us to do this webinar. We're really excited to share this information with everyone today. My name is Dr. Tiffany Wild. I am an associate professor at The Ohio State University in the program of visual impairment education, and my work is on making science accessible to children with visual impairments. Dr. Illich?
Hi, my name is Sonia Illich, and I'm an associate professor and food safety state specialist in human nutrition in the Department of Human Sciences and College of Education and Human Ecology at The Ohio State. My main focus is food safety, and I also have an extension appointment. And with my extension appointment, I do translation of science, and I have done a lot of outreach activities during the pandemic about coronavirus.
On our screen today, on the left-hand side is a tactile model of the COVID-19 that you can use with your students. We're going to be explaining how to actually make that model in just a moment, but what it is it's a picture of an orange with some push pins in it. On the right-hand side of the screen is the picture of the actual guidebook, the Health Education for Students with Visual Impairments guidebook that can be purchased through APH. And what we'll be presenting today will be a module that you can use with that guidebook and the materials found within.
Really quickly, before we keep going forward, I want to remind everyone that if you are going to be applying for ACVREP credit, the opening code for this session is virus. And if you look in the chat, you'll see the link where you need to submit your request for your certificate with the opening code virus, and we'll give the closing code at the end. And just so that Tiffany and Sonia know, the majority, almost half of our participants today are teachers of students with visual impairments. And then another big portion, 38% are O&Ms. Then we have about 6% rehab, vision rehab specialists, about 3% low vision specialists, general education teachers, and college faculty, and students in a college program. We have some of those, some paraprofessionals as well, and then other. And we have people from the Northeast US, Southeast US, about 26% Southeast, 16% Northeast. We've got 25% from the Southwest, 20% from the Midwest, We have some representatives from some of our US territories, Canada, and of course other. So we have a very diverse group of attendees. So we're really excited about that. So I'll be quiet now and I'll let you keep going.
Amazing. Thank you.
Thank you and welcome everybody. And we did this work for our educators and all that work with children with visual impairment. So it's so great to see everyone and to know everyone is excited about learning more about this topic and wanting to know more about this topic. Our goals for the webinar is for you to understand how to create tactile COVID-19 models, to understand the science behind the COVID-19 prevention and recommendations, and to learn how to distinguish scientifically accurate information about COVID-19. We also want participants to understand how to teach about the transmission of COVID-19 and learn how to guide students in performing the experiments designed to teach them about COVID-19 transmission. Our last goal is for you to understand ways in which students with visual impairments can share what they have learned about COVID-19 with others.
So first of all, I have to say that I'm so excited that we have so many participants from all over the US today. And I'm going to start by sharing some important facts about COVID-19. So COVID-19 is a respiratory disease infection that is caused by coronavirus, SARS coronavirus 2. That is the name of the virus. And it has emerged, as most of us know, in 2019, spread globally and caused a pandemic in the US, but in the majority of the world. Coronavirus is not a loner. It has cousins, and it belongs to a large family of viruses, coronaviruses. Similar virus that you might have heard about before are severe acute respiratory syndrome, or SARS virus, and Middle East respiratory syndrome, or MERS virus. And both of those caused some infections and epidemics before. If someone gets coronavirus or COVID-19, that's the disease that is caused by this virus, symptoms include fever, coughing, shortness of breath, other breathing difficulties, loss of taste and smell, diarrhea, vomiting, nausea, but it causes also the range of different symptoms in different people, swelling of joints, etc., a number of symptoms. So, yeah.
Sorry. On the screen, you'll see a photo of two children, a boy and a girl sitting in a gray cloud, and behind them are germs. At the bottom is a resource link for more information about COVID-19 in children, and that resource link should be put into the chat box for reference.
So oftentimes people are wondering if children can get sick with coronavirus. Yes, that they can. They are at lower risk than adults, especially older adults, but they still can get sick with COVID-19. Average incubation period for the kids once they get exposed to the virus is around 7.7 days. It actually ranges anytime from 1 day all to 14 days. And this is why it is important if somebody was exposed to coronavirus to be isolated or in quarantine for 14 days. When it comes to kids, and we know as majority of us here on the call are teachers, kids express more respiratory symptom than adults when they get sick. So there are good chances that they can transmit the disease. In fact, according to this Harvard study, the children contribute to 21% of symptomatic and as asymptomatic transmission. Asymptomatic transmission means that somebody can carry virus, can be a carrier of the virus and be infected with virus and not express any symptoms, but still can infect other people. So who are the people that are at risk from contracting COVID-19? Well, everybody can get sick with COVID-19, except that people with pre-existing conditions can get more severely sick. So people with conditions like various cancers, chemotherapy, different therapies that lower their immune system, diseases, respiratory diseases like asthma, then cardiovascular diseases, even diabetes. And we know that a large proportion of our population in the US has type 2 diabetes. And obviously advanced age is a risk factor for getting severely ill from COVID-19. So sometimes, unfortunately, the exposure and the disease leads to death in these persons. And again, a person can carry a virus and not being sick. So this is why it is very important that we have protective measures at all times.
On
So-
this screen, oh, sorry, on this screen is a picture of the COVID-19 model. It's what a cross-sectional photo of COVID-19. There's genetic material inside the cell, and on the outside are protein spikes protecting the genetic material. These are the receptors of the cell, and this photo was from Scientific American, July 2020.
Yes, so that is our SARS coronavirus 2 that causes COVID-19. And the virus transmits from person to person, mostly through droplets. So when a droplets from an infected individual, when this individual coughs, sneezes, or even talks, the droplets are expelled from our mouth. Even now I'm talking into the screen, and at the end of my talk there are going to be multiple droplets on the screen. So even talking is enough to expel these droplets. The droplets carry the virus. So the person gets sick when the droplets from infected individual land on mouth or nose of another individual, and this individual breathes in the virus. So through the airway, the virus gets to the lungs. It has the receptors on those spikes and attaches to our lung cells and make us sick. So virus can also be transmitted by touching an object, and that's why we focus also on hand washing. So it is really important that we understand that for this transmission of COVID-19, the droplets play a very important role. So the virus transmits from person to person, as we said, through close contact via droplets. Droplets can be large. When we talk, we expel large droplets that travel approximately 6 feet once we cough or sneeze. We also expelled micro droplets, or little droplets that also contain virus, but those droplets can travel much more than 6 feet. They, in fact, can travel up to 26 feet, some studies have shown. This way of transmission is called airborne transmission. And also, as we know, the virus can be expelled from an infected individual onto the surface, and from the surface on will get to another individual by touching. That form of transmission is called fomite transmission. We also worry about that, and this is why we have hand washing. But it is important to understand that the majority of transmission occurs through droplets, either through close contact or through airborne transmission. That's why we say minimum 6 feet, and face masks are critical for prevention. And then obviously hand washing should prevent the fomite transmission through objects.
On this slide, there are 2 photographs. The figure at the top shows the simulation of a cloud of airborne bacteria containing macro drops and micro drops. The droplets also contain viruses if a person is infected. So what you'll see is it looks like a person coughing, and there are rows of Petri dishes showing little clumps and colors of bacteria growing on the Petri dishes. This is a graphical overview of the path of the droplets when there's no barrier or when a face mask is not worn. The bottom figure represents what would happen if people did not keep distance, for instance, in a lineup at a cash register. There are little faces of one person, and there's an arrow there's multiple faces to the right of that with an arrow that says spray reaches less than 180 centimeters with no barrier. If a person at the end of the lineup sneezes, all people standing within the 6 feet from him would be exposed. So in our figure, we are showing 6 people that may be a foot apart in a lineup or any crowding event that could be exposed. There are also 2 links on this slide for more information that will be placed in the chat box. Dr. Elick, do you want to,
Do you want me to take this one? Okay, so this chart represents some of the findings that show the effectiveness of homemade masks. So we were in the lab and we used the simulation that Tiffany showed earlier in the previous slide. So that spray bottle, and we tested how far the droplets travel without barrier, and then with a barrier that is made from different types of fabric. This was at the beginning of the pandemic when we were really concerned that there will be not enough surgical mask supplies for healthcare settings, and people were actually recommended not to wear masks. And we were saying, okay, let's see what is the effectiveness of those face masks that would be potentially made from homemade fabrics. And we tested no barrier. That is in a first group of green, yellow, and white bars that show when we have no barrier, we get a lot of microorganisms, a lot of virus expelled at close distance, but also at far distance. So green, yellow, and blue line bar, they represent different distances. With surgical mask, if you see the next group, the surgical mask is a pretty good barrier and prevents the transmission of the droplets through the barrier pretty effectively, so at any distance. And then the third group represents cotton, that is cotton 140. That would be your cotton that that a normal white t-shirt is made of. And if we put it in one layer, it is going to provide the barrier, especially at further distances, but it's not going to be great barrier. And then the following group represents the cotton with double layer that provides very similar protection or barrier to transmission of viruses like your surgical mask. We also tested other materials like those. We thought, what is that the people have often at their homes, like the kids' old sports jerseys, etc., and they did not provide very good barrier. So it is important what kind of fabric your mask is made of.
Before you move on, there was a good question that I wanted to be able to share with you. Would you be able to describe what the numbers on the y-axis are from that previous slide? Or just review that real quick?
Yes, I'm going to remove the subtitle. Okay, so the numbers on the y-axis are the counts of microorganisms that we counted when we expelled the simulated spray through with the spray bottle onto the Petri plate, as shown in previous slide. Tiffany, can you put the previous slide up? So if you see on the top slide, we have a spray bottle. We are spraying the droplets onto the Petri plate and capturing them on Petri plate. Of course, we know viruses don't grow on Petri plates and we cannot visualize them like this. We use the bacteria for that purpose, to simulate the droplets. We know that droplets from the mouth carry bacteria and carry viruses at the same time. So we're basically able to count droplets. And this experiment is actually the basis for the experiments that are described in the module as well. Is that the answer to that you were looking for?
Yes, I believe that is exactly what the attendee wanted. Thank you very much.
Okay. Thank you. And so I explained what we tested in the lab, and we looked at the effectiveness of different fabrics. But in real life settings, things may be different. This systematic review, represents the findings of studies that were done in observational studies that were done in real life, and were all pulled together and published. The link is available also in our link document. So what they found is the scientists have found 172 different observational studies in healthcare settings, but also outside of the healthcare settings, and they looked at the effectiveness of surgical masks, right? And if you remember earlier, there was a lot of discussion about the effectiveness of masks. And all these studies have been done in 16 countries and 6 continents. So they found, out of these 172, they found 44 studies that directly compared the transmission when you have mask and when you don't wear mask. All right, and all these studies included around 26,000 patients that either had COVID-19 or their cousins, SARS and MERS. And so what they found is that physical distancing of more than 1 meter, which is 3 feet, reduced the transmission by 75%. So no 100%, but for this 100%, we need at least 6 feet, right? And then also the use of face masks and eye protection was absolutely effective and absolutely critical for preventing the transmission. Okay, so we previously worked with the group from Case Western and Kent State University and developed this module for experiments to demonstrate or to illustrate the effectiveness of mask and the prevention of transmission of COVID-19. And this module is published by Frontiers in Medicine. The paper is published by Frontiers in Medicine, and the module is available online for download. We based some of the activities in our modules, we based on the modules published by Echler and her collaborators.
There are 2 websites found on this slide, and they too should be in the chat.
Okay, so we said it is important that we wear face masks, but when we do wear face mask, it is important also that we wear them properly. The face mask, to be effective, needs to fit snugly to your face, and also we said if you are wearing a fabric face cover, it has to have multiple layer, and we demonstrated it in an experiment. We're going to be demonstrating it in the module as well with our students. So it is important that the face mask is always clean and is laundered often. So it's made of a material that can be laundered. If you're having surgical face mask, that face mask is practically designed for one use. When you're eating, when you're doing activities where you do have to remove the mask, it has to be removed properly, making sure that not the inside or the outside are touched with your hands, and the hands have to be cleaned and washed before and after putting and taking off the face mask. For more information on this, there is a website that we included on this slide, and it's in the document as well.
So now we're going to get to the activities that you can do with your students. And these are a teacher-created module that will be available to all of you soon, and we'll give you information about how to obtain this module at the end of the presentation. On the left-hand side of this slide is a picture that was shown on the very first opening slide. It is an orange, so we have the round orange as the cell of the COVID-19 model, serving as the cell. And on the outside are push pins, and these are push pins that I found that came with my bulletin board that I bought as I was creating my office space for working at home. And I put them randomly around the outside of the orange. And those are to represent the spikes, those spikes on the outside of the COVID-19. So that's the homemade version. On the right-hand side, we have a 3D printed version. This 3D printed model is courtesy of Stacy Kelly from Northern Illinois University. It is available for free download from Thingiverse. It is printed in orange because that's the filament that she used. You can use any color filament that you'd like. Again, it is round in the center with the spikes, those spikes sticking out throughout the entire model. So these are two models, and we have step-by-step directions of how to create both models that will be available in the module, as well as the directions to go directly to the Thingiverse site, where Dr. Kelly has placed her downloadable directions for your 3D printer that's available to everyone.
Okay, so now we're getting into activities that are meant to demonstrate the transmission of COVID-19 and the distance to which the droplets fly once they're expelled from a cough or a sneeze. So for this activity, you will need spray bottle, large print or tactile ruler, a writing device to record the result, paper towel, pencils, and optionally a pH quick drop paper. So first, we're going to be demonstrating with the students how far the droplets can travel once they're expelled by coughing, sneezing, or even talking. We will use the spray bottle filled with water, and the water will be sprayed at different distances at a student's leg. So one student will lay on the floor and point the bottle that is sitting flat on the floor at the legs of another student. The student that is standing is then going to step back, make one step back, and the student laying down will spray again. And this process will be repeated until the student who is standing is feeling the droplets landing on their feet. So once the student can no longer feel any water droplets on their legs, the student will measure the distance with a proper measuring device and then record that distance. The teachers should discuss with the students how the droplets felt and with each movement backwards, and what observations could be made and how far the droplets actually landed during the experiment. So in the second part, we're going to test now. Basically, we're going to repeat the same thing, but we're going to angle the bottle up or down. And this is meant to simulate the coughing or sneezing position. So we know that is different if you cough with your head angled down and or with sneezing and coughing with your head angled up. The process will be repeated, just the student who is laying down is going to be angling the bottle up and down. And so the discussion at this point for the teacher with the students is going to be to describe if the flat bottle or angled bottle or bottle angled down or up allowed water to or droplets of water to travel the farthest. So as we said, if the water bottle represented a person sneezing, would you want to be directly in front, angled up or down from that person? And also when you're coughing and sneezing, you should keep your head in a certain position to make sure that droplets are not going far.
An optional activity that you can do with your students may choose to spray the water at different distances onto a piece of APH quick draw paper instead of onto their legs, and then use their hands to feel the swelled paper. Again, students can compare how the paper felt when sprayed at different distances and different angles, and we'll be referring back to this option in one of the future activities.
Yeah, so the results from these activities are basically demonstrating that oral, nasal micro droplets and macro droplets travel pretty far from a person's mouth. And then if we sneeze with our face upward, the droplets, it's going to fly even further. So so from this, we're going to draw the conclusion that we should be trying to look down when we sneeze and cough, and then understanding how far the droplets are traveling. Okay, so we're going to be, the next two activities are going to be about preventing airborne transmission, and then we're going to be talking about the face masks and effectiveness of face masks. And we said the droplets, the prevention goes by via droplets through close contact, or airborne, again, via droplets. So wearing face cover and preventing those droplets from reaching another individual, or preventing those droplets from reaching you is going to be critical. The next critical thing is going to be 6 feet of distance, and understanding how that feels and how we can measure that in different environments. And finally, we're going to talk about how to prevent that fomite transmission, the transmission to the objects and from the objects. And obviously for that, hand washing is the most important prevention tool.
So our activity now is to determine the social distance of 6 feet. This is a great activity for expanded core curriculum. Those of you who are on this call that are orientation and mobility specialists, this would be a great activity to do with your students. Any teachers can do this that's on this call as well. You will need a measuring device, so a large pen or tactile ruler, and a tactile marker. And you can use a rock, you can use tactile tape, anything that will put a place marker on the ground for you. So we're going to start by asking your students, how far away from your own body is 6 feet? And how do they know that? And have a discussion of how they determine 6 feet, since that's so important for the prevention of COVID. We're going to ask the students to stand in a stationary position and tell the student that you will be clapping your hands, and ask them to tell you to stop once the student believes you're 6 feet from them. So the teacher will stand in front of the student, they'll clap, Take one step back, clap, and continue so far and continue that process until the student believes that you are 6 feet away. Once the student indicates that you are 6 feet away, please place a marker on that spot and then measure the distance from the student to the actual tactile marker. If the measurement is not 6 feet away in distance, walk with the student from their starting point to 6 feet away so that they can get an idea of that 6 feet. Then discuss with the student how he or she will figure 6 feet away distance in the future. What markers could he or she use to ensure that people are 6 feet away? For instance, how many cane lengths away should another person be? How many arm lengths could another person be away? Number of steps, etc. How is that child going to determine, or that student or that client going to determine 6 feet away from another person? Then you'll need to practice that 6 foot measurement with the student with the various ideas that were generated to ensure that the student understands scale. And we know this is a tough concept, but it's very important to understand that 6 feet when we're talking about the prevention of the spread of COVID.
So in the next activity is basically the experiment, and we're going to be demonstrating the effectiveness of face masks and exploring the airborne transmission of COVID-19 with and without barrier. And please interrupt me at any time if you have additional questions. This activity is most complex, but it is a wonderful activity that not only showcases the effectiveness of fabric barriers, but also is a full-blown science experiment. So for this, you will need materials: a spray bottle, the same spray bottle that you used in the first activity, or the second activity, measuring the distance, one empty cereal box, 2 pieces of fabric, and this could be any fabric, whether it's old cotton shirt, scrap cloth, another textile that you have available at home or in the classroom. It should be cut in approximately, well, not approximately, exactly 5 by 5 inches. And then you're going to need a pair of scissors and then some object to act as weight. So you will need to secure the cereal box in a standing position by putting in silverware, a soup can, a rock, or anything that provides weight. You will need a large print or a tactile ruler, and optionally you can use APH quick draw paper.
On this screen are 3 photos of preparing the materials for the transmission activity. On the far left is the cereal box, and the front has been cut out on the 5 by 5 inches, as well as the back. So you can see all the way through the cereal box. And there's a hand that you can see all the way through the cereal box. The second picture is a middle photo. It has that cut out cereal box, a spray bottle, 2 pieces of black cloth lying on a table, scissors beside the black cloth, and a roll of red tape. You can use any tape you want. We used red for color contrast. On the right-hand side of the screen is a cereal box. That same cereal box has now been covered with a cloth material and is held in place by tape on all 4 sides.
Okay, so you're going to be securing that box in upright position, as I mentioned, by having objects such as utensils, soup can, or anything that you have handy inside. You're going to be placing this box directly in front of a student that is standing, and then another student will be seated on the floor, holding the spray bottle in upright position, flat on the floor. And the nozzle of the spray bottle is going to go straight through the window. Student will then give one complete spray through the window, and then wait for 30 seconds for the droplets to settle in. The student that is standing in front should describe what they felt once they were sprayed. Then a student who is sitting on the floor who is standing on the floor should step back, and the student with the spray bottle should not move, and then give another complete spray. So this process should be continued all the way until the student who is walking backwards does not feel any new droplets landing on their feet. At that point, you're going to the students are going to measure the distance between the student that is seated and the student that is standing, using the large print or a tactile ruler, and record the distance. And the teacher could discuss with the students at this point how the droplets felt with each movement and what observations they could make from the recorded data. And at that point, they should also be comparing the previous experiment, the previous activity that when students were measuring distance without the barrier.
An optional activity that you can do, again, if the student does not want to spray another student's legs, you may choose to spray the water through the box with a cloth at different distances on a piece of APH quick draw paper, and then use your hands to feel the swelled paper. Again, the students can compare how the paper felt when sprayed at different distances and compare that to the first activity where there were no barriers, and how much of a change occurred from this current spray situation with a barrier versus no barrier.
Okay, so the result, basically the outcome of this activity that needs to be, that will be observed, is that the previous activity without barrier, you're expecting the shorter distance that the droplets are traveling, and you're expecting the smaller less droplets that are going through the barrier. And some of the droplets are kept by the fabric, so that is something that should be discussed with the students at this point. So we said, especially with visually impaired population, they rely on a tactile information much more than the general population. The fomi transmission, or transmission through surfaces, is going to be really important. But as I mentioned before, the fomi transmission is not the highest risk for COVID transmission, and the hand washing is one of the most effective strategies to prevent fomi transmission. Regardless, in the everyday situations, we're going to be in the position to be in touch with many surfaces that are exposed to many people and may contain droplets that carry coronavirus. Especially if, for instance, when we're going grocery shopping, or eating at the restaurants, or taking out food from the restaurants, or ordering the delivery. So it is important when grocery shopping to use hand sanitizer before entering the stores, and then after finishing the shopping. It is important to use sanitary wipes to wipe the cart, wear face cover at all times, and maintain that physical distance of 6 feet. So remember, if you are touching the surfaces, there is a risk of transmission, but you can reduce that risk by washing your hands and using sanitizers. But the greatest risk is to be around people who carry the virus. So when we are talking about takeout and delivery, we have not heard of any reports that people have gotten actually sick by going through drive-through or during the takeout. But to minimize risk, practice hand washing often and use hand sanitizer after hand washing after you handle the packages of the food that were handled by other person.
There is an optional activity that you can complete with your students. It is on page 219 of the Health Education for Students with Visual Impairments guidebook from the American Printing House for the Blind. In this activity, you will use the same spray bottle that's been used in the other activities. And there's a discussion on how germs are spread from surface to surface within that activity. Again, it uses a spray bottle, the quick draw paper, and barriers such as tissues that we use for other diseases. There's a full discussion and additional resources for how germs spread given in the guidebook with this activity.
So the following activity is discussing the effectiveness of face masks and fabrics to prevent transmission of the virus. And you will again use the spray bottle and the cereal box like in previous activity, but there will be some preparation that is involved in this activity. So you actually have to start preparing for this experiment a day ahead of time. You will need some sort of germs. The good germs though, not the ones that cause diseases, but the good germs like the ones that are in yogurt. So you will need either yogurt or kefir or soil or any other culture food that has good bacteria in it or good germs. You will need a cereal box or 2 cereal boxes to be able to do it simultaneously. You will need 6 shallow plastic containers that are at least 2 inch in diameter, so they're round, a foil cupcake liner, a cupcake liner, a paper cupcake liner if it's double, a lid, jar lid, or anything that is round and shallow that you have would do. You will need 1 tablespoon of sugar, 1 cube of beef bouillon or a spoon that is equivalent, and then 2 packages of plain cooking gelatin, water, microwave to heat that all up, and then you will need a Ziploc bag or similar, and some sort of Tupperware or box to store the growing plates. So in this picture, you see pretty much the material that will be needed, and on the left side of the screen is the material: water, gelatin, bouillon, and sugar. If you mix gelatin and water and bouillon and a tablespoon of sugar and microwave that for a couple of minutes until boiled, the mixture will be very similar to the agar that we used in the laboratory to grow the bacteria. So that agar will be liquid once you take it out of the microwave. You're going to pour these plates. In the picture on the right hand side, you see me pouring the agar into the cupcake holders that are actually double paper cupcake holders. Once you pour the plates, they're going to be liquid. So you are going to have to leave them overnight to solidify. You can leave them either in some dry and dark corner that they're not going to be disturbed or exposed to any sort of contamination. For instance, on the top of your fridge, maybe in the microwave if you're not using it overnight. I prepared this last time during the pretty hot summer months, and I had to put it in the fridge. So it is okay to put it in the fridge overnight to solidify. Once we have prepared that, those growing plates, agar plates for germs to grow on, we need to prepare a germ solution. And the germ solution we're preparing on the day of the experiment. So we're basically adding 1 ounce of, which is around 2 tablespoons of yogurt or soil, depending what we have. It is important if you're adding soil to make sure that, that there are no large particles that are going to clog the nozzles on your spray bottle. You're going to be putting that solution into the spray bottle, and you're going to label it as germs. And so that solution, again, contains harmless germs. They're not pathogens. They're good bacteria, good germs, and we're just going to use them to demonstrate the spread of germs without and with the mask. And then in the next part, you're going to prepare your cereal box with window that is 4 by 4 inches wide, and you will need again the fabric that is 5 by 5 inches, new fabric that was not used for a previous experiment or activities. So you will prepare your container as we showed previously. You will prepare your cereal box as we showed in previous activities, but you're also going to prepare a container that is going to be ready to put those plates on once we spray the germs on them. And that container, I use Tupperware container that we're going to see, I think, in the next slide, and a Ziploc bag.
So yeah, is a picture of the prepared cereal box. Again, it has a window that's 4 by 4. A cloth is covering that cutout window that's 5 by 5. And again, red tape is on all 4 sides, holding the cloth to the box, the cutout box. Again, we use red tape for contrast. You can use whichever tape will hold the cloth to the cereal box and cover up the window.
Yeah, so it will be important that while you're doing this is basically microbiological experiment. It is going to be important that you wear your face mask, not to contaminate the germ growing plates, and also not to touch those gelatin agar plates with your fingers, because then whatever germs you have on your fingers are going to grow on them. You're going to prepare 4 of those growth plates, gelatin growth plates, to test the effectiveness of cloth face masks. And we're going to be comparing no face mask with 1 layer fabric with 2 layer fabrics. And then we're also going to be testing the face masks that the student is wearing. So we're going to have 4 items that we're testing in this experiment. So as I said, we prepared already the cereal box with 2 layers. So we're going to test the 2 layers first. We're preparing the cereal box window that is covered with 2 layers of cloth, and we're starting the experiment.
And I want to add here's a tip. You might want to prepare 5 gelatin plates That way you have one plate for the student to explore and to understand the materials that are used in the experiment, to then allow them to touch the gelatin or what would represent the agar. And then therefore they will not, and tell them not to touch the other 4. But then that extra one gives them something to better understand the experiment.
That's an excellent tip. So we're going to turn the box flat so that the cloth covered window is facing up. And then the window that is not covered is going to be sitting on the table or the surface. And then from about 5 inches away from the cloth, at about 45 degrees angle, we're going to spray twice into the window. And then wait for those 30 seconds for the micro droplets to settle, to land, and then carefully take the plate out of the text box of the cereal box and put it in that box that we prepared that we're going to store these germ boxes. At that point, we have completed basically the testing of the two layers of fabric, effectiveness of 2 layers of fabric. Now we're going to repeat the same steps to test 1 layer cloth, and no cover, and the own face masks. We have to remove the cloth from the box, wipe the box to make sure that we cleaned all the debris, and then move on with the rest of the experiment. We're going to use new piece of cloth for 1 layer experiment, and then we're going to be using the plate that we label number 1. So once we have sprayed the germ solution from the spray bottle on plates number 1, 2, 3, and 0, we're going to put all those in the germ box and the growing container. We're going to cover it with a Ziploc. We're covering it with a Ziploc because we don't want it to dry overnight, because these plates are going to have to be incubated for 24 to 40, more likely to 48 hours. So we don't want them to get dry. So it is important that this is covered. We want to store the box with the gel plates, growing plates, in the place where they're not going to be disturbed. The place that usually works for me is on the top of the fridge or any sort of cabinet that is out of reach, and we'll make sure that this also area is pretty warm. The warmer the temperature, the faster your germs will grow.
So what we have pictured here is what Dr. Illich described before. So on the left-hand side is a picture of the cereal box with the window displayed on top. And she is placing one of the cupcake liners with the agar solution into the box. The second photo on the left, the cupcake liner is already slid into the box, and a spray bottle is on top of the cloth covering, spraying the solution, the germ solution that's either made of the yogurt or the soil. Again, further illustrating the experiment, on the left-hand side is a picture of 4 cupcake liners that have already been sprayed. So the 4 cupcake liners with the agar solution that represent the no cloth, 1 layer, 2 layers, and the child's face mask. All 4 of those agar plates have been placed into a Tupperware container. That Tupperware container is then being slid into a bag And this is a Ziploc bag with a seal on it. On the right-hand side is a preview of what should happen at the end of this experiment. After the 24 to 48 hours that you have left the plates in a warm place, such as on top of the refrigerator, you'll see little white things growing or little dots growing on top of the agar plates or your cake liners.
Yeah, so at the end of experiment, I know we're pretty short on time, but just to emphasize, we need to really clean out, dispose of the box, and clean the working area, and incubate the gels, the gelatin plates for about 24 to 48 hours. Remember to always have clean hands and wear a face mask when you're checking the plates. What maybe we should, Tiffany, can I get the next slide?
The way to observe your Petri dishes, we want to bring in again the expanded core curriculum and self-determination. So one of the things you can do is have your students use the Be My Eyes app or the Aura app if you have access to that service, to have the person on the app service describe the image on the Petri dish. Another optional way to complete this observation is to take a photo of each dish, print the photo onto capsulated paper, and place the paper into a pee-off machine for an instant tactile graphic of each of the Petri dishes. Or you can have a peer or a teacher could describe the image to the student, and that observation is baseline data. You'll want to make these observations in 24 hours and 48 hours using the same method that you chose before. You'll want to record the observations for how many colonies were on each plate and what was the area of the plate. They can measure that if that's appropriate.
We're a minute over time, so you might want to wrap it up. Yeah, thanks.
So yeah, so have discussions with the students, and also you'll want to share that knowledge with others to make sure that they have shared their learning with others. We'd like to acknowledge those that made the citizen science activity possible for us. And the access to this whole module with all the lesson plans will be made downloadable from the APH shop site, and a blog will be written to announce when it is ready for download. We have resources and references for you in the chat, and we have questions if anyone would like to stay and ask. Our contact information, I'm at wild.13@osu.edu, and Dr. Ehrlich is ILIC_2@osu.edu. Thank you.
Thank you, everybody. We'll be happy.
Yeah. Go ahead, sorry.
We'll be happy to answer any questions either now, if somebody's interested to stay longer and ask questions, or at any time, just send us an email, please.
Yes, and for those who have to hop off, the closing code for ACV REP credit is the word clean. So for ACV REP, the closing code is the word clean, and it's been put in the chat box already as well. And if anybody has any questions, sounds like Dr. Ehrlich and Dr. Wild are willing to hang on for a minute.
Yes,
yes. Your question in the chat. No questions yet? Just lots of accolades. Thank you for a great presentation. Thanks for all the ideas. Thanks for all the resources.
Thank you, everybody for listening and for coming to hear this webinar.
Yep. Oh, the closing code is the word clean. Somebody asked for that. Sorry, go ahead, Tiffany.
Oh, I just want to thank you for allowing us to be part of this series and for presenting this information. And we're so excited, want to get this out into all of your hands, and let us know how it works with your students. If you have any comments or questions as you go along, we're here to help as well.
Definitely.
Yeah, we're thankful for you guys too, because this is so important, and just knowing how to make this accessible for our kiddos who, you know, who normally wouldn't be able to access the information in this way. That's a very important thing that you guys are doing, and I really appreciate that. And I know APH is really happy about that as well, and all the people who attended are really excited. All the wonderful comments coming up. I think there was a question about whether or not we'll be sharing this, and yes, it is being recorded and it will be shared on our APH YouTube channel. And then I believe the PowerPoint, the link to the PowerPoint was I see it in the chat right now. Amy just posted it again.
Just dropped that in the chat. And Belinda, thank you for captioning. We can let you go, Belinda. Have a great day over in Germany, and we will continue doing our own questions here.
Thank you, Belinda.
Thank you. And I also wanted to say, please share this also with your gen ed teachers. This is fully accessible for everybody and a great unit for everyone. So please share with your teachers and your colleagues across the educational field.
Yes, and then citizen science module is also available, and the link is in this PowerPoint and in the additional document that we send with all the resources and links. So for the modules for the students without visual impairments. So please do share. I think that it is amazing that this module is available to download as a free access, and that makes me really happy. Which in any case, if anybody has any question and if anything is not going as it should be, just let us know.
There is a question from Joanna in the chat box that I just want to make sure we can circle to. I know, Joanna, that you've been patient. You had asked that in the beginning, and when I saw that come back the second time, I was cognizant of it. She wants the clarification on the cereal box. Are both windows on the cereal box covered with the cloth, or is there only one window covered?
So that's a great question, Joanne. That only one window is covered. So when you're covering, we had to make two windows just to have things go through the box. But only one window is covered, whether with one fabric or double layer fabric. And basically, when you're putting that box on the flat surface like the table, you're putting a cupcake holder or a gelatin plate right into that other window that is not covered. Does that help?
She writes, thank you very much. She couldn't tell in the picture how the box was covered, so thank you.
Thanks for the question, Joanne.
All right, let's see. I couldn't, okay, all right, there we go. Joanne was just saying thank you because she couldn't tell in the picture how the box was covered. So that's good. That
Thanks.
was helpful. Okay. Oh, the capsulated paper you mentioned, Tiffany, is this Sewell paper or Sol paper?
It's the capsulated paper that you use to put into the pee-off machine.
Okay. Okay, she acknowledged that. Acknowledge your answer. That's good there. Somebody had a question about ACV REP credit. I'll get that up there for them. Okay, any other? Nope, I don't see any other questions coming in so far. Oh, okay. Yep. No other questions. It looks like we're winding down our the numbers. We went from 200 down to 44 now. So it's like a little at a time. People are leaving as they've gotten their questions answered.
Yeah, just let us know. It
Yeah.
was really a pleasure to work with this team and with Tiffany to put this together. And we're very happy to share any knowledge and that we may have that can help.
Yeah.
Thank you all so much.
Yeah. Okay, Amy, I think we're good.
I think it is good. You can go ahead and close things up.
Okay, thank you so
Thanks,
much.
everyone. It was great.
Thank you, everybody. Thank you.
Okay,
Thanks,
everybody stay
Tiffany.
safe.
Thanks. So long.
Bye-bye.
Bye.
We'll be in touch, everybody.
Yeah, I'll be in touch with you.
Bye. Thanks so
Bye.
much.